just happened here? Okay.
The meeting is recording.
It used to tell us when it was
I guess
recording.
I'll begin then, yes?
Yep, you can go ahead, Jim.
Okay. Hello. I hope everyone can hear me. This is Linnea Skoglund at Montana State University. And we've got Jim Stack chomping at the bit to get started on his presentation today. So I think I'll let him give whatever introduction about himself he would like to give. And we're anxious to hear about this wheat blast. Jim, go ahead.
Well, good morning, everybody, and thank you for your patience as we worked through a long list of technical difficulties beginning about 2 days ago. So hopefully this will go okay. If not, we'll fix things up for the recorded version that gets archived. I do want to mention that I see from the list of participants, we have two of the experts on wheat blast. I'm not saying that in jest, that's in earnest. Bill Bacchus and Christian Cruz have done a lot of the work that I'm going to discuss today. So hopefully they can correct any errors I might make or chime in with additional details, or certainly answer questions at the end. So let me begin. Wheat blast, an emerging disease in South America. What I'm going to try to do over the next few minutes is give you an overview of the disease itself, but also specifically information about the projects that have been underway here in the U.S. and in South America to address this emerging disease. Well, why do we even care? Of course, everybody knows we have a food security challenge that we're facing. We're going to add another 2 billion people to the planet over the next 30 years. And here in the U.S., we're going to add another 250 million people to this country this century. So we have to maintain the capacity to feed all these folks. FAO, the Food and Agricultural Organization, says we need to increase food production by 70% over the next 25 years. And that 90% of that increase will come from the intensification of existing production systems. Currently, we have about 250,000 plant species that have been described or predicted. Over 30,000 plant species are considered edible. About 7,000 plant species are used for food, and about 120 plant species are currently cultivated. However, the advance isn't working. Let me see if I can get this right here. However, just 4 plant species provide 59% of the calories that we consume. 11 plant species provide about 93% of the calories that we consume, and those are the same 11 plant species that we've been consuming for about 10,000 years. Now, importantly, wheat provides about 17% of the calories in the human diet. Wheat is grown on more acres globally than any other crop, so anything that threatens wheat production is of high significance. Wheat is a critical food crop to address global food security. What I'm not going to discuss today are some other projects we have here at K-State looking at food security and trying to identify critical areas in the world that are vulnerable to some of the challenges to plant health. One of the areas of concern, of course, is Asia, and I'm just going to use India as an example. If we look at the current situation, India is the second most populous nation in the world, second to China. However, projections are that sometime around 2025 to 2030 timeframe, India will surpass China as the most populated nation on the planet, reaching about 1.4 billion people. And the significance of that is that in 2013, they're just barely food secure, and wheat is an important commodity to them. So if we look to the future, sorry, I'm still having a lot of difficulty getting things to advance here in the animations. If we look to the future with respect to climate change, we know that Bangladesh is going to lose more land, and India may become too hot for wheat. Well, that's pretty significant. If we look at wheat blast as a potential threat to wheat production globally, some projections have been made as to where wheat blast may get established and where it might cause damage. And again, if we look at India, and the slashed areas are the very high risk areas. India, again, if climate change doesn't knock wheat out, wheat blast might. So wheat blast might be a significant issue for us to address. If we look at what we know about other populations of this pathogen, rice blast, for example, we know that it's a global threat to rice production, and that the management of that disease has been problematic. We've been breeding for resistance to rice blast, but the average rice cultivar lasts only about 3 years before the pathogen population adapts to it. So we've got a lot of work to do to understand better this wheat population and to see just how similar and how different it might be from the rice population. Clearly, it presents a challenge. There are many populations of Magnaporthia rizae, the pathogen that we're talking about today, but they do cluster into different phylogenetic groups, and interestingly, the wheat population seems to be more closely related to the ryegrass pathogen than it is to rice, and the supposition is that the wheat pathogen did not actually evolve from the rice population, contrary to some early belief on that a few decades ago. Well, currently, wheat blast is in South America. It's in Brazil. It's in Bolivia and Paraguay. It was first detected in Brazil in 1985, and by 2013, it's in Brazil, Bolivia, Paraguay. Probably had an outbreak, at least a detection in northeast Argentina a few years ago, and a detection in Uruguay, but doesn't seem to have been established there. So it's in South America. It has spread from its original outbreak site. Currently, hundreds of thousands of hectares are no longer planted to wheat in Brazil, primarily due to wheat blast. And Brazilian farmers are currently using a wheat blast forecasting model to demonstrate their risk in order to get loans to buy wheat seed. So this is pretty serious in South America. Now, wheat blast in the U.S., has it ever occurred? Well, I think it was 2012 in Kentucky. There was one wheat head in a breeder's plot that had wheat blast symptoms. But what was determined based on whole genome sequencing was that that particular population that hit the wheat emerged from the lolium pathotype, the ryegrass population, and was not related to the wheat blast populations from South America. It's based on fairly good data. We have a couple of the isolates from South America fully sequenced, and there are isolates of the ryegrass population in the U.S. that are sequenced, and the evidence would suggest that that one occurrence in Kentucky was not a derivative of a population from South America. It hasn't been reported since, and there's no indication that it's spread. Having said that, this is a very interesting species, Magnaporthia rizae, that seems to be able to throw out variants fairly regularly. So we probably need to keep an eye on the lolium population as well as what's going on in South America. What does this thing look like? Well, let me acknowledge right now Guillermo Berea, Diego Valdelamar, Christian Cruz, Bill Bacchus, and several others for providing images to use in this presentation. So it does produce leaf symptoms, but not regularly. It's believed, yeah, it's the animation. It's believed that the conidia produced in these lesions, whether it be on the wheat in the field or whether it is in grass hosts at the margins of fields, can disperse and cause head infections. And that's the most significant aspect of this disease, are the infections of the head. And you can see there are these point lesions. And what happens, depending upon where that infection occurs on the head, it can basically kill the rest of that panicle, rachis. Oh, man. However, again, depending upon the presence of the pathogen and the weather conditions at the time of dispersal, you can get isolated infections, and so they can be dispersed throughout the wheat head instead of from a point and then up. When it gets established, it can cause devastating disease. Entire fields can be affected by this pathogen. That's one of the interesting aspects of the epidemiology to be determined yet, is exactly where does all this inoculum come from that seems to result in a somewhat synchronous outbreak, but a lot of details missing on the epidemiology of this disease. The impact, of course, is you either get no seed or the seed is of very, very poor quality. Interestingly, some of the outbreaks, some of the epidemics they've experienced in Brazil, you can get head blast without a lot of leaf symptoms. So again, the question is, where does that inoculum come from to support that magnitude of infection in the field? Now, we don't know how much of the knowledge that's been generated for the rice system that can be applied to the wheat yet, but we're working on that. But if we look at this, it may be a combination of grass hosts on the margins of fields or in the region. It might be volunteer plants left behind from the previous crop. It could be seed that was left on the soil surface from the previous harvest. All of those are potential sources of inoculum to support these large-scale epidemics. And this was from an epidemic in 2009 in Brazil, and all of that is not mature wheat, that is blasted wheat. There are some resistant wheat cultivars, but they don't appear to be high yielding. They don't appear to be completely resistant to the pathogen. And the experience to date in both Bolivia and in Brazil is that chemical control is not fully effective. Now, whether that's a function of the chemistries being used or the application methodologies are not mature is again not completely clear. I will say that the Brazilians have been working extensively on this. There is a lot of literature in Portuguese about wheat blast. We've been able to translate some of that. This is the Royal We, Christian Cruz, and a few other folks on the project have been able to translate some of that to make that information available. The symptoms, they can be confused in the field with other diseases. So you look at the healthy neck there and a neck that's diseased with wheat scab. That's blast. And that's physiological browning that occurs with some cultivars. The symptoms on the leaf can vary depending upon the host. And again, there's a lot of confusing literature in Brazil as to the host range for this particular pathogen. Magnaporthe oryzae is isolated frequently from quite a variety of grass species, but whether all of them are capable of causing wheat blast is not quite clear, so that still needs to be sorted out. This is a wheat head affected by tacol, by blast, and by scab. And so you can imagine that with the variation you would expect across different cultivars and different environmental conditions, that in the field, unless you're an absolute expert with this, it could be very easy to make a mistake with these symptoms, either overlooking it and thinking it's scab, or mistakenly believing it's blast when in fact it's one of these other diseases. There are some resistant varieties under development, and you can see that the differences are quite dramatic, but there is nothing approaching immunity in either the Bolivian or the Brazilian breeding system. So, I mentioned it was originally detected in Brazil in 1985, and it began to spread. And get into Bolivia and into Paraguay. But in the previous decade, there have been some very dramatic epidemics. In Brazil, it's believed in 1 year, I believe it's the 2009 epidemic, that the combination of wheat blast and head scab reduced the national yield by 30%. In individual fields, blast is capable of upwards of 90%. I mean, we're talking devastation levels when this disease gets roaring. And so, of course, it's caught the attention of a lot of folks, wondering whether this pathogen is going to get dispersed around the globe and what impact it might have if it does get dispersed to other areas. So in 2008, a group here at K-State partnered with the Agricultural Research Service at Fort Detrick, scientists there, partnered to get a grant from the AFRI program, and that partnership also involved bringing in scientists from Brazil, Bolivia, and Paraguay. That project was successful. We got a lot done, and then in 2012 we received another grant, new grant for 5 years, 5.1 million, I believe it was. And in that, we have now 5 land-grant universities involved. It's Kansas State University, the University of Arkansas, University of Kentucky, Ohio State University, and North Carolina State University, as well as the ARS lab at Fort Detrick. And once again, we have scientists in South America fully engaged in this project. And you'll see why that's so important to have those partnerships in South America. The upper group is the full blast team. The lower group, in addition to the people I mentioned, USDA APHIS, Center for Plant Health Science and Technology, is involved in this project, and I'll discuss their role in a few moments when we talk about the pathway analysis that was done largely as part of Christian Cruz's PhD thesis. So Christian's been providing a lot of the leadership, and he does all the work, and we basically take all the credit. This has garnered worldwide attention, and Brazil hosted an international meeting in May of 2010 to figure out if we can design a mechanism that allows us to fully collaborate, meaning share resources, sending isolates from one lab to another, conducting tests for each other in different locations to see if we can't get a fast start on addressing this new pathogen. It was organized by Embrapa Wheat, Embrapa Cerrados, and CIMMYT. And out of that meeting came the establishment of a consortium. It's basically a contract signed by 11 different countries, I think 13 institutions, and that may be growing. I think that actually has grown a little bit. So there's worldwide attention on this new disease, a lot of interest because wheat, again, as I mentioned earlier, is grown on more acres than any other crop, and it's considered one of the critical crops for addressing food security. So what are the questions that we're asking? Well, some of the obvious ones. Will it spread? Will it spread within South America? Will it get into the United States? Will it go global? What impact will it have if it does spread? So in the U.S., is U.S. wheat susceptible? In South America, will it continue to spread throughout the region? And then, of course, globally, what impact will it have if this is able to jump continents? Well, one of the most important aspects of working with an exotic pathogen is biosecurity. I'm preaching to the choir here, but it's important to walk through this. So if you're going to work with an exotic pathogen that poses, well, even if you're not sure, but if it has the potential to pose significant threat to a major commodity, then it's incumbent upon the researchers to do that in a manner that's going to preclude it escaping into the natural setting. So biosecurity is fundamentally about two things. It's about containment and it's about exclusion. And that concept applies across all scales, whether you're talking about a field, a geographic region, or a facility, or even a lab. So we want to ensure that that organism can't escape, and that means you have to develop protocols that are specific to that organism. And for the most part, for most of the work we do, the organism is contained within a Class 2 biosafety cabinet. And so we're not talking about growing this up in 55-gallon drums or anything like that, or using it in the field. This is all the work done in the United States is done at either the Biosecurity Research Institute at K-State, which is a BSL Level 3 facility, or at the Biocontainment Facility at Fort Detrick, Maryland. But we do want to inoculate plants, and so Christian Cruz and Bill Bacchus devised a way to inoculate plants that minimizes the inoculum that passes through. So if you look here, this is an inoculation chamber. We have a HEPA filter at the back of that chamber that's sucking air in. And so the airflow is going into that filter. And when you then spray those plants with your inoculum, what is not deflected or what is not impacted onto the plants goes into that HEPA filter. So that really minimizes the amount of dispersal within the lab. I'll just take a brief minute here. A lot of effort went into working out safety protocols and security protocols for this laboratory. So experiments on how long will a conidium survive if it lands on a lab bench or on the floor. What is the proper chemical to use? Do you use bleach? Do you use any of the other disinfectants that are on the market? How effective can autoclaving be? And what are the specifications for that autoclaving with respect to the moisture of the material or the duration of the temperature maintained? All of those things have to be worked out. Some of that work was done with a surrogate, with the Lolium pathotype, and then the rest was done after we got the pathogen into the building, done with the real organism. Sorry that I'm still having a little trouble with the animation. So, laboratory has the entire laboratory air is filtered through a HEPA filter prior to exiting the facility. So even though we are working within a Class 2 biosafety cabinet and we have a special inoculation chamber and it's a shower out lab, so we have to don special clothing, shoes, and protective equipment, as you can see Christian here inoculating plants, to ensure that we're not going to exit with any of these conidia on us. And then we shower out every time we exit that lab. So this is a highly regulated process. All soil, plant materials, potting materials are placed into autoclave bags. The bags are sealed and the outside of the bags are disinfested. And then they pass through the shower block before they go to the autoclave, which is all still inside of containment. So all solid waste is autoclaved before leaving containment. All liquid waste is autoclaved before leaving the facility as well. There's a central waste decontamination plant in the basement of this building. So that's what we do to ensure biosecurity when we're working with it here in the U.S. in the lab. But what about field biosecurity? What about when you're working on this organism in another country? What do you do? Well, we have to worry about a couple of things. That when you're out working in the field, as most of you have known that have done field work know, that you can't help but pick up soil and maybe plant materials and smears on your clothing and boots. And so we had a rigorous policy on how we decontaminate, how we address that. We brought autoclave bags with us. Our clothing went into the autoclave bags, got autoclaved on returning or bleached. We laundered clothing before we left the country. And we scrubbed boots. As you can see here, we're spraying them down with Clorox solution, scrubbing to make sure in the sole there are no trapped soil particles. We then stored all of those boots on-site, in-country. They did not leave the country, the field test sites. Now the samples that we wanted to come back with had to be treated under protocol from APHIS, under permit. We had rigorous protocols, as you can see here. I think some animation's missing, but basically we packaged any plant materials in Ziploc bags with desiccants, and then those bags were placed in another container with additional desiccant. They were in basically FedEx type packaging to be rapid delivered, and you can see all of them had the approved APHIS labeling. And again, you can see we leave the boots and some of the clothing left behind. So with the laboratory biosecurity concerned with containment and exclusion, you have to establish protocols that are specific to the organism and to the experiment. So it's not just the organism, but what are you going to do with that organism? So all the protocols revolve around that. And then you follow those established protocols. The field biosecurity, with basically 3 general principles. Don't bring anything with you. So just as much as we don't want anything getting into the United States, we shouldn't bring anything with us when we go to another country. So you need to think about how to minimize that risk. So don't bring anything with you, don't spread anything around while you're there, and certainly don't bring anything back. And we had worksheets, work lists, and behavioral lists that we followed in order to accomplish that. So what did we do in this project research-wise? Disease phenotyping led by Bill Bacchus and Christian. That work was done at BRI, the Biosafety Level 3 lab I just mentioned to you. Again, it has its own shower-out block right from that laboratory. Fort Detrick, and over 500 wheat cultivars have been screened to assess their susceptibility and resistance to this pathogen. Field experiments in Bolivia, we had some significant challenges. I'll show you some of that in a minute. But a basic agreement of results, nothing to indicate a major conflict. But we have an awful lot of work to do on the field side for validation of what we're learning in the laboratory. So how does this phenotyping occur? As I mentioned, the inoculations occur in this inoculation chamber. They're placed into a growth chamber. Now we have 2 growth chambers inside of our biocontainment laboratory. So once these plants are inoculated, they never leave that laboratory until they're bagged in autoclave bags and ready for autoclave. About 14 days after inoculation, they're screened for disease type, disease reaction. One of the important things to do initially was to determine if there was any correlation between the seedling resistance susceptibility to the mature plant head infections and susceptibility readings. That's important because if you could use the seedling, then it's a whole lot easier to do this work. Bottom line is they did some preliminary experiments at BRI and looking for correlation between seedling stage and head stage. And there was a correlation, but the decision was that that correlation was not significant enough to rely on your seedling scores as predictors of adult plant resistance. And so all subsequent screening was done with adult plants. So mature plants with the head emerged from the boot. This is an array showing the disease severity ratings for 85 wheat varieties. This is work done by Bill Bacchus and Christian Cruz at the BRI. And you can see quite a range of disease reactions, all the way from very minimal disease response to the full head being blasted. And so it's encouraging in the sense that there may very well be some resistance that becomes usable for breeders. That work can be seen in its entirety in 2 publications, Cruz et al. 2012, Plant Disease, and Bacchus et al. 2012, and wow, the rest of that just disappeared. It's somewhere down below that slide. I'm not sure what just happened, but it's in plant health progress. Wow, what just happened? So I just lost my entire screen. Give me a second here. Okay, well, so plant disease management reports. It was an animation issue. This is one of the field test sites in Bolivia. A beautiful place to do work. These are our field plots. So we set up some experiments with collaborators in Bolivia to see if the results we were obtaining at the BRI would be predictive of field performance. It was a serious challenge, and if we go back and we look at this here, I'm not sure what you can see or not, but okay, this is one of the first tests we've done, and you can see we didn't exactly get a uniform stand of wheat. It took us a while to figure out what was going on. Is it's a different latitude and different day lengths at the time of testing. And the problem for that is wheat is day length sensitive and will not mature fully develop actually if it doesn't receive the right number of hours of light during the day. So in a subsequent test, our collaborator in Bolivia grew up plants in pots in a greenhouse and then transplanted them to the field and installed lights to try to provide the adequate amount of lighting to get these plants to fully mature. And I'm just kind of going through all this to give an idea of what it takes to get some field work done. Our results are pending. In Bolivia, we're also doing some scouting to see if we can identify the reservoir hosts for this pathogen, the plant species that will support this population in the absence of wheat. Christian has been collecting plant materials in different locations and mostly based on those that are showing symptoms that could be Magnaporthi, and we're trying to type those eventually. So that's what we've been doing here. Now, one of the other aspects is trying to figure out what's the likelihood that this thing could actually leave South America and get into another country, specifically the United States. The United States grows a lot of wheat, but it's also a very important exporter of wheat, and that's important with respect to the global stores of some of these basic commodities. We looked at the occurrence of wheat blast in Brazil, and what you see on this slide, on the left, PR is the state of Paraná, and on the right is the state of Rio Grande do Sul, and those two states have the pathogen Magnaporthi oryzae triticum pathotype. So they have the wheat pathogen well established. So the pathogen survives very well in both locations. However, wheat blast epidemics only occur in Paraná. And so it is a good opportunity to look at the different conditions in these two states and see if we can identify differences that would help to explain why we get epidemics in one state and not the other. So we looked at accumulated infection days during the wheat growing season prior to heading. We looked at accumulated infection days during wheat heading period. And I'm sorry, I'm going to go back here. What you can see is that there are differences, and it's in the northern part of Paraná. I'm sorry the animations aren't working effectively. In the northern part of Paraná, where the epidemics are most severe. And you can, if you look at, if you compare those two states, you'll see that the weather conditions are in fact different. Less so for infection days during the heading period. So this is when the most severe part of the disease is happening. And the differences between the two states are less dramatic, less apparent. And then if we look at the accumulated days with daily temperatures falling below 0 degrees during the wheat growing season, you'll see that the two areas are dramatically different. Now, why this is important I'm going to mention in a few minutes, but we're looking at the ability of this pathogen to survive between wheat crops. So we're comparing that, and then we're looking at what's the likelihood that this pathogen is going to get out of Brazil. So we're considering two possible vehicles for that. One is, could it spread naturally? Meaning, could it spread throughout South America, maybe through Central America and into the U.S.? And we know there's a continuous wheat belt from Mexico right on up into Canada. So either spreading by successive waves in a standard epidemic process, or through the benefit of a hurricane, much like Ivan introduced soybean rust into the southern United States. Could that be a vehicle? So that's one mechanism. Christian Cruz focused on trade as a possible vehicle for his thesis, and that's what I'm going to mention here. So I'm not going to go into any detail here, but the bottom line is, we asked the question, could infected kernels from Brazil end up in the United States? And just a brief overview, it involved developing a lot of probability analyses for different stages of that process. So, for example, what's the probability that there's an epidemic in the state of Paraná, Rio Grande do Sul? Because those two states provide 90% of the wheat that gets exported from Brazil. So we know the pathogen's established in both, but what are the probabilities of an epidemic? And then what are the probabilities that that wheat ends up in export? How many kernels, if you looked at the incidence of infection using some basically means of prior experience, you come up with some estimation for the percentage of the kernels that would be infected, and then based on the magnitude of wheat exported, you can actually come up with numbers of kernels infected that would be exported. Now, what Christian learned during his research was very interesting, that there's actually a program in Brazil to help farmers get the maximum value from their production. One way to do that is to put your product on the global marketplace, export it. So exports have a higher value than using it domestically oftentimes. So what happened in 2009, as I mentioned earlier, is there was a serious epidemic of wheat blast in Brazil. That's the one where they attributed about a 30% loss nationally to wheat blast and to scab combined. So that was a big epidemic. In order to, so the wheat from that affected area was of very low quality. The PEP program stepped in and encouraged them to export that wheat so that the farmers would get more money for their crop. So what we know is, and that occurred, those exports occurred in 2010 and 2011. So what we know is that there was an epidemic in Paraná in 2009, and in 2010, some of that wheat was exported. So first question was, what's the likelihood this could get out of Brazil? And the answer then is, yeah, it can. Can it get out of Brazil? Yes. So the next question then is, can it get into the United States? Again, Christian learned some very interesting things during his research, one of which was that wheat is not just a food crop for humans, it's a feed crop for livestock and poultry. And there was a cooperative established in North Carolina amongst poultry and swine feedlot operators to purchase grain on the open market, the global market, because the value of the wheat in the U.S. was high, and they could purchase it less expensively offshore than within the U.S. So they actually built an elevator in Wilmington, North Carolina, to receive imports, and they purchased some of that wheat from Brazil in 2010. So we know that MOT-infested seed was exported in 2010 from Brazil, and we know some of that wheat was imported into North Carolina in 2010. Now, that doesn't mean that the pathogen was. It doesn't mean that we've had an incursion. We don't know that. We're just looking at overall behavior and estimating the possibility that these events may have occurred. So can MOT get into the U.S.? Yes, it probably can if it gets in through these trade channels. But will it spread? Will it get established? These are basic questions within South America, looking at into the U.S., globally. We talk about these are our basic questions. So if it gets into the U.S., what's the impact going to be? Well, we know some of our wheat varieties are susceptible. I showed you data from Bacchus and Cruz earlier. What's the possibility that this will get established? So Christian spent a lot of time developing some models, and this is where we collaborated with scientists at the Center for Plant Health Science and Technology in Raleigh, North Carolina. Roger McGarry, in particular, was the leader on that side, but there were some other scientists as well involved. And if we look at this graphic, this is the y-axis is the proportion of winter wheat production area. So it's a proportion from 0 to 1. And then on the x-axis, we're talking about the probability that MOT will establish. What we can see here is that for 60% of the winter wheat areas in the U.S., the environment is not suitable for MOT establishment. So for most of the winter wheat areas that you see shaded brown, MOT is not likely to get established. What you can see here in the colors is the frequency of occurrence of climatic conditions that would allow establishment. So the red area means that 10 years out of 10, if it gets in, it'll get established in that location. Whereas the dark blue would be 1 year out of 10, it might get established. But what about an outbreak? So that's saying that it can get in and become resident in that habitat. What about an outbreak, a disease outbreak? And again, we're looking at the same y-axis, the proportion of the winter wheat acres affected. And then the x-axis this time is the probability of a wheat blast outbreak. So this is disease. And what we see here is that for over 70% of the winter wheat acres in the U.S., the environment is not suitable for a wheat blast outbreak. So most of the acres won't support establishment, and most of the acres will not support an outbreak of a disease. And what we're going to look here at the same type of plot, where the brown shaded areas are where winter wheat is grown, and then the different colors represent the number of years out of 10 that the climatic conditions would support a wheat blast outbreak. And what we can see is that it's really only this small area in south central U.S., part of Louisiana and maybe Mississippi, where the possibility of wheat blast occurring is significant. So it's a very small area of the U.S. that appears to be, and again, I want to stress, all of this is preliminary until we get more information on the biology and the epidemiology of this pathogen. We had to operate on a lot of assumptions because data are missing for MOT. One of the things Christian did was to look at other populations of Magnaporthia rhizi, so the rice population, MOO, as well as the ryegrass population, MOL, and see where they occur in the U.S., see where they survive. And based on some data generated with the Lolium pathotype publication, Harmon and Latin in 2005, Christian generated a probable survival map of MOT in the U.S. So everything in red is where red means 10 out of 10 years the weather conditions would preclude survival. So the wheat blast pathogen would not likely survive in those areas of the United States during the wintertime. And then it transitions into areas of the United States where it might very well, where the conditions would not affect its survival. So can it get into the U.S.? Yes. And, oh, sorry, we decided to, animation. We decided to get a little more specific and address specifically that possibility for North Carolina. And we developed a corridor approach for assessing risk. And it's, again, based on that target area in Wilmington, North Carolina, or basically southeast North Carolina. So now we're looking at, and you can see the heading, wheat production areas at risk, North Carolina. So the y-axis, proportion of winter wheat area, same as in the previous graphs. And the upper graph here is the probability that the wheat blast pathogen will become established. And again, this is southeast North Carolina. And what you see here is that in 7 of 10 years, 60% of the winter wheat area will support the establishment of the wheat blast pathogen. Now what that looks like is this. So these are the counties that we considered, and this is based on tracking down where those feedlots were located. And this is using GIS analyses. We were able to track down where the feedlots were located. We were able to locate where wheat is grown. Christian used the most probable routes of travel from the port of entry to those feedlots, and using estimates from the rice blast pathogen on dispersal capability, we determined if the pathogen was to disperse from that corridor, how far. And that told us basically it can disperse about 1,000 meters. So that meant that on either side of the transportation or the road, you would have a corridor of about 2,000 meters wide, a corridor around that road. Christian did a lot of work interacting with experts in the grain trade and learning that the estimates are that when grain is moved from one location to another, they estimate they lose about 1% to spillage. So when that truck is driving from the port of entry to the feedlot, grains are falling off that truck. And most of us know that work in the Great Plains, where we're out driving around the Great Plains and you see volunteer wheat, volunteer sorghum on the sides of the road. Those are grains that fell off the trucks on the way to market. So we use that as a probable dispersal mechanism that these grains would fall off the trucks on the way to the feedlots. And all the statistics I mentioned earlier about the proportion of kernels infected and their survival based on the maps that you saw. So using dispersal capabilities, the proportion of kernels that are likely colonized by MOT, and the survival, and all of that generated a risk that told us what the likelihood is of whether this pathogen would get established should an infected kernel fall off a truck in southeast North Carolina. And that's what you see here. Again, these risk rankings over on the extreme right are telling you in basically how many years out of 10 the conditions would support establishment. And you can see it's significant. A good part of that is at least 5 years out of 10 conditions would support the establishment of the pathogen. Well, what about an outbreak? So that's saying what you're seeing in the upper graph and on the right is that the pathogen can get established. But could it cause disease? Would the conditions favor disease? So the same type of analyses were performed, and what you see here is that in 1 of 10 years, 50% of the wheat area will support, well, I'm sorry, that should say outbreak, and a wheat blast outbreak. And this is what the map looks like. So although the pathogen would get established, the conditions would support establishment at least half the time, it would be rare that the conditions would support an actual outbreak of the disease. And so even in this targeted area where we think there's a mechanism to allow the pathogen to get introduced, the likelihood of a wheat blast outbreak appears to be very low. Again, stressing that we don't know a lot. I can see I'm almost out of time. There are a couple publications coming out on this. I'm going to move quickly. I just realized the time. The new project involves several institutions. We're going to be doing more phenotyping, developing a forecasting model, an MOT-specific diagnostic protocol, and continuing to work in South America. And with that, I will end. Sorry for taking so much time, and I appreciate your patience with all the technical difficulties. Have a great day.
Thank you, Jim. Do we have any questions? Anyone that's got a live mic, or if you wanted to type in a question there? So it looks like up here in Montana we're sitting pretty sweet. We're not going to have to worry about this one, huh?
Well, that's the guess. You know, that's the best guess at this point. Yes.
We'll keep the stripe rust, and you can have the wheat blast.
We have stripe rust as well.
Yeah, that's true. You do.
I'd be particularly interested to hear Christian or Bill Bacchus if they saw anything that needs clarification in that. Actually, I don't see Bill any longer. So, Christian, if you're still on and you have anything to add, please do.
Can you hear me?
I guess not.
Okay. Well, I'm sure you can.
Christian, go ahead.
I just wanted to mention, stress out a little bit that.
No, I think that was just me.
No. Actually, you can't hear him, Linnea, but he's speaking.
Yeah, I just
go ahead, Christian.
Just wanted to say that one of the biggest questions that we have at this point is, where is this inoculum coming from? And as Jim said, it's possible that it's coming from the host itself, and that's something that we are doing research now here at the BRI. There's some preliminary data that I would like to present, but pretty much we're seeing symptoms on the leaves at this point. So that's something to keep in mind.
Thanks, Christian. Anyone else? Again, sorry for going so long.
Okay. Evidently, I'm not hearing everything, so if somebody's trying to talk, just tell me to hush up.
I'm not seeing any chat, and I'm not hearing anything. We'll try to clean this presentation up before we archive it. I'll try to fix the issues. Well, thank you for your time, everybody.
All right. Thank you, everybody. Sharon? My computer's choking again. If you can hear me, would you please shut down the